The difference between bioluminescence and fluorescence. Bioluminescence and fluorescence are two fascinating natural phenomena that involve the emission of light. While they may seem similar at first glance, they are fundamentally different in their mechanisms, sources, and applications. Understanding these differences provides insight into the intricate workings of nature and the ways organisms use light for survival and communication. What is bioluminescence? Bioluminescence is the ability of living organisms to produce light through a chemical reaction within their bodies. This process involves specific molecules: luciferin, a light-emitting molecule, and luciferase, an enzyme that facilitates the reaction. When luciferin reacts with oxygen in the presence of luciferase, light is emitted as a byproduct. Bioluminescence is often referred to as cold light because it produces very little heat, making it an energy-efficient way for organisms to glow. Key features of bioluminescence: internal light production. The light is generated by the organism itself through a biochemical reaction. Energy source: the energy for the reaction comes from ATP, adenosine triphosphate. The cell's energy currency: self-sustained, low. Organisms do not require external light sources to produce bioluminescence. Purpose: biolofactory, such as attracting mates, e. fireflies, luring prey, e. anglerfish, or deterring predators, e. some squid release glowing ink. Examples: fireflies, jellyfish, anglerfish, glowworms, and bioluminescent plankton. What is fluorescence? Fluorescence is the emission of light by a substance or organism that has absorbed light or other electromagnetic radiation. Unlike bioluminescence, fluorescence is not a chemical reaction within the organism. Instead, it is a physical process where an external light source excites molecules, causing them to emit light. How fluorescence works. A molecule absorbs light energy, usually ultraviolet or blue light. The molecule enters an excited state. As the molecule returns to its stable ground state, it releases the absorbed energy in the form of visible light. Clearly, emitted light is often a different color from the absorbed light, depending on the properties of the fluorescent molecule. Key features of fluorescent molecule. External light source needed. Organisms or materials must be exposed to an external light source to fluoresce. Instantaneous reaction. The light emission occurs only while the external light source is present. Energy source. Fluorescence relies on photons from the external light source, not chemical energy within the organism. Purpose. Fluorescence is typically a byproduct of a molecule's structure and not necessarily used for survival or communication by the organism. Examples: corals, jellyfish, some species, certain minerals, and fluorescent dyes used in science and medicine. Interactions. Organisms that blur the lines. Interestingly, some organisms exhibit both bioluminescence and fluorescence, depending on their biology and environment. For instance, jellyfish. Some jellyfish are bioluminescent, producing their own light through chemical reactions, while others fluoresce under ultraviolet light due to fluorescent proteins. As diamond-thin corals fluoresce, creating vibrant underwater displays when exposed to ultraviolet or blue light. These overlapping phenomena demonstrate how nature uses light in versatile and complex ways. Applications of bioluminescence and fluorescent bioluminescence. Applications: medical research. The luciferase enzyme from fireflies is used in cancer research and to track cellular processes. Inheritance monitoring. Bioluminescent bacteria are used to detect pollutants in water. Biotechnology. Bioluminescence is used in genetic engineering and drug testing. Fluorescence. Applications. Molecules. Fluorescent dyes help scientists visualize cells and molecules in biology and medicine. Forensic science. Fluorescent materials are used to detect biological traces at crime scenes. And design. Fluorescent pigments create vibrant visuals under UV light. Conclusion: bioluminescence and fluorescence are two distinct yet equally mesmerizing phenomena that showcase nature's ability to manipulate light. While bioluminescence involves internal chemical processes to produce light, fluorescence depends on external light sources to excite molecules. Both play vital roles in science, technology, and our understanding of the natural world. From the glow of fireflies in the forest to the vibrant fluorescence of coral reefs under ultraviolet light, these phenomena highlight the diversity and beauty of life on Earth.